Welcome back to all our viewers not on campus with us today on our Facebook live stream. We are back with our final segment of Alen Deluxe, and don't forget that if you are around with us or are still making your way to campus, we have our closing ceremony at 4:30 between the EH Little Library and the Lilly Gallery. There will be free cake at 4:30 p.m., so stop by. With me right now, I have the Davidson College Collegium Musicum, who are a madrigal group, an offshoot of the Davidson College Chorale, which is the premier audition ensemble for voice on campus. So without further ado, I'm going to hand it over to them to share a piece of music with us.
Now is the month of May, when merry lads are playing. La 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 la, la 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 la. Now is the month of May, when merry lads are playing. La 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 la, la 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 la. Each with his bonny lass, a dancing on the grass. La 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 la. The spring glad all in gladness doth laugh at winter's sadness. La 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 la, la 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 la. The spring glad all in gladness doth laugh at winter's sadness. La 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 la, la 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 la. And to the bagpipe sound, the nymphs tread on the ground. La 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 la. Why then why sit we musing, you sweet delight refusing? La 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 la, la 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 la. Why then why sit we musing, you sweet delight refusing? La 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 la, la 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 la. Say dainty nymphs and speak, shall we play body brave? La 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 la. Say dainty nymphs and speak, shall we play body brave? La 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 la.
Thanks, you guys. That was awesome.
Thank you.
Good luck with the rest of your set. Have a great day. Once again, that was Collegium Musicum, an offshoot of the Davidson College Chorale, and a whole bunch of good friends of mine. So glad they let us stop by for a song.
Awesome. So we're going to walk down the natural science poster fair is being set up right now. And so we're going to try to find someone who we can talk to. I think we have someone right here. How's it going? Could you talk to me about a little about your research, Ella, and what you've been doing?
All right, so I looked at single photon interference, which can occur when a photon is sent through an interferogram, so it has a choice of two different paths, and the photon comes in with a polarization of 45 degrees, and one path has a 90-degree polarization and one path has a 0-degree polarization, so it can go either way easily. And after the interferometer, you place a quantum eraser, which sets the polarization back to 45 degrees, so the photon cannot remember which path it took. And consequently, it thinks it went both ways at once and creates an interference pattern with itself. So it behaves like it was two photons.
Wow.
So that's what you see there. And we can tilt one of the beam splitters in the interferometer to create a greater path length difference between the two paths. And that creates the sinusoidal pattern. And so in these plots, we're looking at how the number of coincidences between the photons hitting our two detectors at the same time changes as you change the path length difference, as you make that greater. And as you can see, when we have a wider range of wavelengths coming into the interferometer, the amplitude of the interferogram decays more quickly, and when we narrow the range in wavelengths, the decay is slower, and so you can see that it's more gradual on that plot. So yeah, this is our relationship between the wavelength range and the coherence length, which is the distance between half max amplitudes. Yeah. So yeah.
This is so beyond me. I took AP Calculus in high school, and that was about it. So phenomenal work. Thank you so much for talking to us. And so we have over here some more physics presentations. we go. Just walking a little ways here. Awesome. And how's it going? Well, if you could speak to us a little about your project this semester.
Yeah, so I'm in one of the genetics labs on campus, and we're looking at a protein that when it's mutated, it causes infertility in fruit flies, which is an organism that is really similar to humans in terms of DNA. So our findings can probably be applied to human research and treatment methods in the future. So yeah, we're studying a specific mutation and kind of figuring out the mechanisms behind that and kind of how, like, potential treatment options and how it works.
What's the mutation? What does it cause exactly?
So the mutation is in this protein called NMD, which is expressed throughout the body. It helps with the health of mitochondria, but the mutation that we're studying only causes flies to be infertile. So it's only affecting their reproductive system. So we're kind of figuring out why that happens, why the mutation's not affecting other organs, and kind of the consequences of that. Yeah.
Awesome. Thank you so much.
Yeah, no problem.
Awesome. So we're going to be showing one more presentation here on the second floor. How's it going?
Hi, how are you?
Good. If you could speak a little bit about your project that you've done this semester.
Oh, okay. So this is kind of the culmination of all our ideas from our Health and Human Values class, Health, Genes, and the Environment. And so we focused on the issues that smoking has in utero, based on your epigenetics, which are the proteins and everything that are along the genome. So it's phenotypic changes without any type of changes in the genome. So, and we focused and found that smoking causes issues along the lines. So, if a pregnant woman is exposed to cigarette smoke, then not only is the child that she's carrying affected, but also the next generation. So your maternal and paternal grandparents can cause issues for you.
You would never think that. You think what you're doing right there just affects you and yourself only, and maybe secondhand smoke too. But the fact that your grandchildren can be implicated in that is really fascinating. So thank you so much for that research.
No problem.
Awesome. So we're moving downstairs, and just as we've seen physics and genetics and health and human values, I think that really speaks to the interdisciplinary nature of this building. The wall space, not only did Jacob take a political science course in here, as he mentioned earlier, but you have physics courses, you have natural science courses here, bio and chem. You have neuroscience laboratories in here too. So the students are doing research that they're performing hand in hand with a professor here. And so I think just walking around here right now and seeing all these different types of projects in the different disciplines, I think it really speaks to the incredible nature of this building that we're standing in right now.
Even now, we're walking through the main atrium where we have our giant display screen where there's a original film project being displayed that was created by a senior, Paul Brennan. This, again, just shows you how multi-purpose this whole building is, as well as how interdisciplinary a lot of the approaches to learning are at Davidson College and in this new and absolutely gorgeous facility. So, thanks, Alex. I hijacked the mic from you there.
No. So we're gonna speak to a few more students here. 1 second.
Right here, Alex.
Yeah. How's it going, all? Could you speak a little bit about your projects that you've been working on so far this semester?
Sure. So the project that I'm working on is basically looking at the structure-function relationship in a certain protein that's a membrane transporter. And the reason why it's important, the main reason, is because membrane transporters are really hard to work with in the lab. And because they're so hard to work with, they're kind of underrepresented in the literature. So studying this will contribute to a growing body of knowledge. So, yeah.
Thanks, man. Yeah, all this science is just right over my head. They signed the arts majors on to come do the talking bits, but this stuff is just utterly fascinating, and it's so cool to see undergraduates being able to engage in this kind of research and have a really hands-on role beyond just a more, abstract intellectual engagement with the material. They really do get to get their hands dirty and experience the science working with professors and other students. So this is absolutely amazing stuff. Let's see if we can butt in here real quickly.
Right. So this was. Oh, sorry.
Cypria, can we knife in real quick? Can you just explain your research real quickly to us? We're live with the Davidson Facebook page right now.
Oh,
so
cool.
just
Yeah.
folks to share some of what they've been doing. Sorry to scare you.
No, you're fine. Okay, so I'm a genomics major, and so I did my thesis through the center. And what I did for my thesis research was I analyzed ovarian cancer samples and the RNA sequencing data that came from them. And so my research culminated in several genes of interest that were most enriched and most differentially expressed in these pathways, which sounds maybe confusing, but it just means that these are the genes that were found to be different between ovarian cancer samples and normal cancer samples. And basically what I found was just these genes are differentially expressed, and this is how they interact with each other, and ultimately I found the spindle assembly checkpoint pathway to be enriched, if that means anything to you. But yeah.
Wow, that's so cool. Well, thanks for mentioning too, or sharing your self-design major with it. I mean, it's cool that folks get to make their own major and then also get to do awesome research on top of that it's not just locked into one track. So thanks for chatting with us. All right, so yeah, again, that was Cypria. She is a senior, and she designed her own major for the Center for Interdisciplinary Studies, and then had the ability to, from there, engage in research from there. So it's there are some schools where you can design your own major, but then who knows, you might be trapping yourself or cutting yourself out from a lot of these resources. But Cypria's gotten to kind of forge her own path, Davidson, which is pretty cool. So I think we're
almost done here.
we've got one more. We got one more. Silly me. Here, I'll pass it on back.
No, yeah. Rebecca Strawn is here. She's a senior with us, and she's going to speak a little bit about her research or her thesis that she's been doing the entire semester. And no, this year actually. I'm sorry.
Just the semester.
This semester.
Yeah, actually, some of my projects are being displayed at the digital fair that's happening at 3:30. So if anybody's on campus and wants to stop by, it's a really exciting part of the digital studies department, which is actually a really new branch of campus. We just got a new lab in the VAC building, so we've been taking advantage of a lot of really awesome resources that we just got access to this year, so make sure to stop by.
That's awesome. Thank you so much.
Thank you.
So that about does it for Lenda Lux so far. If you're still on campus and around campus and you can make it to the closing finale around 4:30, please do. There will be cake, as we mentioned, in the Richardson Plaza right there between the library and Chambers. Again, share your moment at #LendaLux, please. And we hope to see you soon. Thank you so much.